So friends, we were discussing about the steps that are involved in a plant tissue culture experiment. We have already discussed the cleaning and sterilization of the glass and plasticware. Then we have discussed various components that are used or the components of a culture media. So depending upon your requirement, you select the appropriate component and then you prepare your media. So coming on to the next step, that is the preparation and sterilization of the culture media. Now when you have selected the required components from which you are going to prepare the media, you can prepare in two ways. First one, you can weigh the required amount of every component every time and then mix it. And second method is you can prepare the stock solution of each component and you use the required volume of the stock solution in the preparation of the culture media. Now coming on to the first method, wherein we weigh the required amount of each component every time and then we mix to prepare the culture media. Now this method or this way of preparing the culture media is very much cumbersome, because every time you have to weigh each component in the required quantity and then you have to mix it. And when you are going to weigh each component every time in the required quantity, which is sometimes very small amount, there are more chances of error in weighing those components and preparing the culture media. So the end product would be the culture media not of the appropriate quality, and therefore the second method, that is we prepare the stock solutions of the required components and we use the required volume from those stock solutions to prepare the culture media, is more preferred method, and it is less laborious and less time consuming method. Now since the preparation of stock solution is always preferred, so how to prepare the stock solutions? Now we discussed in the components of the culture media certain macronutrients and certain micronutrients, or minor salts or the major salts. So if we are preparing a stock solution of a major component or the major salt or a macronutrient, then the stock solution should be prepared in a concentration 10 times than the required concentration. Likewise, if we are preparing a stock solution of a minor salt or vitamin or a micronutrient, then the stock solution should be stock solution of such salts or ingredients should be prepared in a concentration 100 times that of the required concentration. Now while preparing the salt solutions or the stock solutions, the solution of each salt should be prepared separately and then they should be mixed in appropriate concentration. Now before mixing different salt solution, one has to ensure that the salts that have been added should get completely dissolved into the media before they are being mixed. And once they are mixed, another precaution one has to take is to see whether any kind of precipitation should not occur while mixing the two salt solutions. So to overcome such kind of precipitation problem, it has always been recommended that you should dissolve the inorganic nitrogen source salts first, then we should add phosphate or calcium salts. This will prevent the problem of precipitation. Now if we have to prepare the iron solutions or the ferrous salt solution or the iron salt solutions, ferrous sulfate and sodium EDTA solutions, since it's a major salt, are prepared in a concentration of 10 times than the required concentration. These two ferrous sulfate and sodium EDTA solutions are prepared separately and then they are gradually mixed in the required amount. The pH of the final solution is then adjusted to 5.5 and final volume is made up. If you get a deep golden yellow colored solution, that will indicate a proper preparation of ferrous sulfate salt solution. In order to prepare the stock solution of plant growth regulators, they are normally prepared in a concentration range of 1 millimolar or 10 millimolar. Cytokinins are normally dissolved in 0.5 normal HCl and the final volume is made up and then the pH is adjusted to 5. Unlike cytokinins, oxins are dissolved either in 95% ethanol or 1 molar potassium hydroxide or 1 molar sodium hydroxide and the final pH is adjusted at 5. Likewise, oxins, gibberellic acids and abscisic acid, they are solubilized in 95% ethanol and their pH is then again adjusted after making the required volume. Once the stock solutions or the salt solutions or growth regulator solutions are prepared, then each component according to the list is mixed. Now, many a times it has been observed that scientists used to mix the solutions of various components and during the course of mixing they forget which solution they have already added. So, to overcome such kind of error, it is always advisable to prepare a list of the component and then add the solution of each component and check it off on the list. So, in order to overcome any kind of error of forgetting what type of salt solution have already been added to the mixture. You must take care that each ingredient must be completely dissolved before the next solution of the salt or growth regulator is added into the media. So, if you are preparing 1 liter of media, it is always advisable to start with 400 ml volume of water in a 1 or 2 liter beaker which is placed on a stirrer and then you gradually add the required components into that 400 ml of water. Again, it has been seen lot of errors occur while measuring the volume of the required stock solution or the solution that have to be added to the culture media. It is always advisable to use the pipettes of appropriate volume to pipette out the required amount of the stock solution. Say for example, you want to pipette out 0.5 ml or 1 ml of the stock solution. So, it is not recommended that you should use 10 ml pipette for pipetting out 0.5 ml or 1 ml of the stock solution. To pipette out such small amount, you require 1 ml or 0.5 ml pipette with maximum graduations in order to reduce the error while measuring the volume. Once all the ingredients have been added and dissolved, then the sufficient volume of water should be added, keeping in mind the amount of the gelling agent that has to be added so that the final volume should not cross the required volume. So, in simple words, we can say the sufficient amount of water should be added so that to keep the final volume below the required volume. Once this has been done, then the pH is adjusted with 0.2 molar potassium hydroxide or 0.5 normal HCl to the required value as per your as per the requirements of your study. Now, the volume of the culture media is brought to the final volume, again keeping in mind the amount of gelling agent to be added. Once all the ingredients are added, last ingredient is the gelling agent, as we discussed, that should be added while continuous stirring in order to avoid any kind of clumping. The gelling agent should be added in appropriate amount in order to avoid any kind of vitrification in the media. So, sufficient volume of the gelling agent should be added and finally the final volume of the culture media is made up to the mark by adding the base of the media, that is water. So, once your media is prepared, next step is to sterilize that media. To sterilize the media, autoclaving is the method of choice, and we have already discussed that autoclaving at 121 degrees Celsius at a pressure of 15 psi for 15 to 20 minutes is sufficient to completely sterilize your media. But if you are using larger volumes, so depending upon the volume you are using, you have to adjust the time period of sterilization. Now, if you have already added sugars to your culture media, then the longer duration autoclaving should be done only after prior testing. So, we take small amount of culture media and we expose that small amount to the longer durations of autoclaving, say 40 minutes, and if the media after 40 minutes of autoclaving is clear, then we can subject the entire culture media for proper autoclaving till 40 minutes, because it has been found that if sugars have already been added to the media, they may undergo caramelization, which will destroy the entire culture media when no prior testing is done. And the second method to overcome this is we sterilize the sugar solution separately and then we add into the culture media before autoclaving. Certain heat sensitive constituents are also present in the culture media, like certain vitamins are there, gibberellic acid is there, or any other growth regulator is there. So, if such kind of components are to be used in culture media, they should sterilized by filter sterilization using appropriate pore size filters which can filter out all the microbes, and you get a sterilized solutions of such heat sensitive ingredients. And once these are filter sterilized, then they are mixed into the main culture media under sterilized culture media under laminar flow, keeping in mind the final volume of the media. Now, in this table you can see the effect of the sterilization time as well as temperature on different contaminants. It has been found that most of the vegetative cells can be deactivated when they are sterilized for 5 to 10 minutes at 60 degrees Celsius. Whereas for fungal spores or yeast spores, if you want to deactivate them, then 50 minutes of sterilization at 80 degrees Celsius temperature is sufficient. When you sterilize for 5 to 10 minutes at 60 to 80 degrees Celsius, streptomyces spores are completely deactivated. Bacterial spores in general, they get deactivated at when you sterilize at 121 degrees Celsius for 5 minutes. But there are certain species of bacteria like Bacillus stereothermophilus which do not get deactivated when you sterilize it for 5 minutes. So, for such kind of spores to deactivate, you have to sterilize the media for 15 minutes at 121 degrees Celsius. So, this is how, depending upon the contaminant which may be present in your culture media, you have to select the sterilizing conditions. Now, sterilization can be done by two methods. That is, first we pour the culture media into the culture vessels and then we sterilize them all together. This is the first method. And second is, we sterilize the media separately and then we pour the sterilized media into the non-autoclavable pre-sterilized containers. So, second method sometimes tend to cause further contamination. Therefore, first method is largely preferred, wherein we pour the culture media into the desirable vessels and then along with the vessels we sterilize the entire components. So, coming on to the first scenario, wherein we pour the culture media into the final vessels and then we sterilize. So, the media is poured into the desired culture vessels. Generally, if we are using test tubes, then 25 into 150 mm glass tubes are used, in which we pour around 15 ml of the culture media, or if we are using 250 ml flask, then around 50 ml of culture media is poured. If during pouring the media it starts to get solidify, then it is always advisable to reheat the solidified media on a water bath or in a microwave oven, and then you pour it only when it is uniformly in the liquid state. Now, once you have poured the media in the desired vessels, the mouth of those vessels should always be covered with the non-adsorbent cotton which is wrapped in the cheesecloth. Now, this closing with the help of cotton will allow sufficient amount of the gaseous exchange in the culture. Or apart from this non-absorbent cotton, you can always use any other suitable closure that you have in your laboratory. Now, the culture vessels containing the media are then transferred into the appropriate baskets covered with the appropriate material, aluminium foil or any other material, which in order to prevent the wetting of the closing plugs or the closures of those vessels during the autoclaving, and then they are autoclaved for the specified period of time under specified conditions. Now coming on to the second scenario, wherein the culture is sterilized separately and then it is poured into the pre-sterilized non-autoclavable containers, and this is largely done whenever we are using non-autoclavable plastic culture vessels like vials are there or jars are there or petri plates are there. The media, appropriate volume of the media, say it is being sterilized in 250 ml or 500 ml flask with suitable closures, and once they are sterilized, then they are transferred into the aseptic area where they are allowed to cool down, but they are allowed to cool down only up to a temperature above their gelling temperature, because after that they will again get converted into solid. So say for example, if you are using agar as gelling agent, so you will allow that media to cool down at a temperature to say 50 degrees Celsius, where it will remain in the liquid form, and then you can easily pour into the final culture vessels. Now whatever culture media you have prepared after sterilization, if you are storing it under refrigeration, then you can use that media within 1 week. If you are not storing that media under refrigerated conditions, then you have to use that media before 1 week, because it may start getting degraded. It has been found that if the media is stored under non-refrigerated conditions, the pH of the media drops over the period of time, and this is largely due to the production of organic acids into the media. Now whenever culture tubes are being used, it is always advisable to form the slants of the media, and these slants have been found to provide larger surface area for the growth of the callus cultures. So these are some of the precautions or some of the methods by which you can sterilize your culture media. Various commercial mixtures of culture media, pre-sterilized commercial mixtures of culture media are also available in the market, like MS media or Gambox B5 media, and these commercial mixtures have certain advantages over the lab prepared culture medias. There are reduced chances of errors while weighing the components of the individual components of the media. If you are preparing a culture media in the lab by weighing individual components of the media, there are more chances of error while weighing them or while pipetting out the required volume of the stock solutions. In comparison, if you are using the commercial mixtures of the culture media. Now these commercial mixtures can be stored in less space for longer duration of time, rather than storing the containers or the jars of each component, which will require large space. We just require less space in storing the commercial mixtures of these culture media. Now since now we have prepared and sterilized the culture media, so the next step is the selection and sterilization of the explant. Now explant is largely defined as the piece of the plant tissue or piece of a plant part that is used to establish the culture. Now it can be a part of the leaf, it can be part of a bud, it can be part of a stem, it can be an anther, it can be a pollen, it can be part of a root, it can be part of a rhizome, or it can be small fraction of a callus that is grown under aseptic condition. So anything which you are using, whether from the plant or from the artificially grown plant tissues, is known as explant. Now there are various advantages of using explant from the plant or the culture growing under controlled conditions. Such kind of explant material are free from any kind of infection and they are more homogeneous in comparison to the plant part that is being used as an explant. Now, there are various factors that govern the selection of an explant material, like physiological or ontogenic age of the explant. It has been found that younger the explant, more the chances of its proper growth. It has been found that the younger plant part have cells which grow rapidly, which have higher capability of division, cell division, in comparison to the cells of a plant part which is obtained from an aged plant. Therefore, age of an explant is an important factor while selecting. It is always recommended to select a young explant material. Second factor is the season in which explant is obtained if you are collecting that explant directly from the plant. It has been found that plant have maximum capability of cell division during the spring season, and under extreme summers and under extreme winters they enter into a dormant stage. So if you are collecting your explant in winter season or in extreme summers, then obviously they will have less capability of the growth in comparison to the explant that has been collected from the plant material during the spring season, where they have full capability to flourish. Size and the location of the explant also determine whether the explant will have proper growth on the culture or not. Larger the size of explant, more energy storage will be there in the cells, more cell division will be there, and better growth will be there in comparison to the smaller size. Location of the explant also determine its capability. If you are selecting the tip of the leaf, again we know that tips always have a have the cells having high division capability. If you are using the apex of the stem, obviously they will have cells having higher cell division capability. So such kind of explants will perform better under the aseptic in vitro conditions. Quality of the source plant, whether the source plant from which you are getting the explant is disease free or not, whether the source plant is having proper growth or not. So all these factors will also determine whether the explant will show proper growth under in vitro conditions or not. Plant genotype, again it will determine it will affect the quality of the source plant. And last but not the least is the ultimate goal of the cell culture. What type of culture you are going to perform will also govern the selection of the explant. Like if you want to go for clonal propagation, then lateral and terminal buds are normally preferred. If you want to go for callus culture, then you can grow callus culture from almost any kind of explant material. It can be a cotyledon, it can be a stem, leaf, embryo, any kind of explant can be used for the callus culture. If you are going for protoplast isolation, then normally leaf tissue, specifically from aseptically germinated seeds, are considered as good source. For anther and pollen are normally used for haploid plant culture or the callus cultures. If you the main goal of your culture study is for the genetic transformations, then leaf or nodal meristem or root tips are the choice of explants. So depending upon the goal, you have to select the explant material. Once you have selected the explant material, next step is the sterilization of the explant. We know that plant tissue culture, I again repeat, that is a technique where utmost sterility or aseptic conditions are required. So if the explant material from which you are going to develop the cultures is not properly sterilized, the whole exercise will become futile. So in order to sterilize the explant material, sterilizing agent as well as the duration for which you have to expose the explant material to the sterilizing agent will depend upon the type of the explant you are using. Chlorine bleach, which is normally known as sodium hypochlorite, is the most widely used sterilizing agent, and it is largely used in a concentration range of 3 to 10% in water if you are using the commercial bleach solution. If you are using pure sodium hypochlorite solution, then a concentration of 1% in water is normally used. Apart from chlorine bleach, ethanol 70%, calcium hypochlorite along with 0.5% HCl, hydrogen peroxide in the range of 3 to 10%, mercuric chloride 0.2 to 0.5%. Mercuric chloride is largely used for highly contaminated sample, or you can also use silver nitrate in a concentration of 1%. Now sterilization, as we discussed, that sterilizing agent as well as duration of sterilization will depend upon the type of the explant. So friends, in my next lecture we will discuss about the different plant parts that can be used as an explant and what are their sterilizing conditions that are required. Say for example, if you are using small kind of seeds, what type of sterilization conditions you have to use. If you are using aerial parts as an explant, so different sterilization conditions are used. If you are using roots or rhizomes which are highly contaminated with mud or soil, then what kind of sterilization conditions are required for such samples. So we will discuss in our next lecture. Thank you.